Hi, everybody. My name is Megan Hopper, and today I'll be talking about chemoreception, which is how our body senses chemicals, like how we pick up smells in the air or tastes in food, and then turns that into signals that our brain can actually understand. The two important chemical senses in chemoreception that we'll be discussing today are smell and taste. We'll discuss them in order. So, first, we'll talk about the sense of smell, moving on then to the structure of olfactory neurons, then taste and taste buds, and finally, some interesting facts to just tie it all together. Starting off strong with the sense of smell. Sense of smell is also known as olfaction, and it's the process of using the nose to sense millions of different odors in our environment. This process is going to be beginning in the olfactory epithelium, which is a thin lining inside the nasal cavity. This region is packed with olfactory sensory neurons, the specialized cells responsible for detecting odor molecules. These neurons are highly sensitive and allow us to distinguish between a wide variety of smells, from freshly brewed coffee to cigarette smoke. The axons of these sensory neurons bundle together to form the olfactory nerve, which is also the cranial nerve 1. These axons then share information with secondary sensory neurons located in the olfactory bulb, which is a structure that sits at the base of the forebrain. The olfactory bulb is going to then process and organize the incoming signals and refine them before passing them on. From here, higher order neurons are going to carry that information from the olfactory bulb along the olfactory tract to the olfactory cortex of the brain. One unique aspect of this pathway is that it actually bypasses the thalamus altogether, which a lot of other senses, in fact, most other senses, rely on as sort of a relay station. So, this direct route helps explain why smells are so strongly tied to emotions and memories, since the olfactory cortex connects very closely with the limbic system. So, the overall order of smell processing, just as a recap, is going to start with the olfactory epithelium, then move on to olfactory sensory neurons, then to cranial nerve 1. From there, it'll move to olfactory bulb, and then higher order neurons are going to head over through the olfactory tract and then the olfactory cortex. So, now that we've seen the big picture of how the smell pathway works, let's zoom in and take a little more close look at the makeup of olfactory neurons themselves. So for this part, you can refer to this image over here on the left. So the makeup of olfactory neurons is what we're looking at. Olfactory neurons are actually quite specialized and very unique compared to other sensory cells. So each neuron has a single dendrite that extends from its cell body into the olfactory epithelium. At the other end, there's a single axon that projects to the olfactory bulb. These neurons are unusual because they only last about 2 months before dying off, but don't worry, they regenerate continuously, which is something that most neurons in the nervous system actually can't do. At the surface of the olfactory epithelium, the dendrites end in knobby terminals that connect to non-modal cilia. These cilia are coated in a layer of mucus, which is secreted by Bowman's glands. And when odor molecules enter the nose, they dissolve into this mucus layer and then penetrate it. And then they can bind to olfactory receptor proteins on the cilia. So, this is going to trigger a signal transduction cascade, which is eventually leading to the brain perceiving the smell. Each receptor responds to only a limited range of odorants, but because humans have hundreds and hundreds of receptor types that are all different, we can perceive actually millions of combinations. So that covers how our smell receptors are structured and how they work. So now let's switch over to our other major chemical sense, taste. Our sense of taste is also known as gustation. This begins when a substance dissolves in the saliva and mucus that is within our mouths, and this allows molecules such as sugar, salt, or acids to interact with taste receptor cells. Taste receptor cells are also known as TRCs, it's shortened that way. So if you see a TRC throughout this presentation, that just means taste receptor cell. On the apical membranes of these cells, there are proteins that act either as receptors or channels. So when a taste molecule, also called a tastent, binds to one of these proteins, it starts a signal transduction cascade. And this cascade is going to end with the release of chemical messengers that activate sensory neurons, which send information to the brain where taste is perceived. So, kind of a similar manner to smell, but not quite. Taste is actually essential, not just for enjoyment, but also for survival. So, for example, detecting bitterness can protect us from poisons, while detecting sweetness can actually help us seek out energy or carb rich food. This gives us a basic idea of how taste begins, but taste actually happens through specialized structures called taste buds. So, now let's jump over and take a closer look at that. So, taste buds are small structures found mainly on the surface of the tongue, although a little known fact is that they can also be found in other areas like the roof of the mouth and the throat. Each taste bud contains 50 to 150 taste receptor cells, and they play a major role in determining and differentiating flavor sensations. Contrary to popular belief, this was actually dispelled a while ago. It was once thought that different sides or different sections of the tongue detected specific flavors. This is actually not true, as you can see from the image on the left here. So this is what a taste bud looks like. So every individual taste bud has all of these different sections within it, and all of them will detect various flavors. So it's not just one side of the mouth or the tip of your tongue that tastes sweetness or anything like that. It's actually every taste bud is going to sense those tastes. So, humans are generally able to detect five basic taste sensations, and these are going to be sweet, sour, salty, bitter, and umami. Umami is the one that people don't know about as much, and umami is going to be the taste sensation that comes from amino acids, and this is often described as the savory taste or like a meaty taste. So, foods that are like soy sauce or Parmesan cheese or mushrooms or broth or something rich like that are full of umami flavor. So next time you're eating something with mushrooms, you can know that your umami portion of the taste bud is being activated. Within each taste bud, there are four main types of cells. So type 1 cells, these are support cells. They may also help detect the flavor of salt, although the jury's still out on that one. We're not really sure if that's true or not, but not really sure where the sense of taste of salt is coming from. Type 2 cells, these are receptor cells that are going to detect sweet, bitter, and umami flavors. Type 3 cells are presynaptic cells that respond to sour tastes. Type 4 cells are precursor cells that can develop eventually into new taste receptor cells, more TRCs. So type 1, 2, and 3 are all going to be taste receptor cells, and type 4 is going to be a precursor, so that's a basal cell. Every TRC is going to be a non-neural polarized epithelial cell. What does that mean? That means that it's not a neuron itself, but it works closely with neurons to pass along taste signals. And these cells are actually going to be found deeper in the epithelium. And the only part that actually reaches the surface of the mouth is the taste pore, which is where tastins enter. A lot of people think that those bumps that you see on your tongue are taste buds. They're actually millions and millions of, if that's too many, then hundreds of taste buds around all of those little bumps that you see. So they're really microscopic. You cannot really actually see them with the naked eye, but there's a lot of taste buds on there. So to summarize, type 1, possibly salt. Type 2, sweet, bitter, and umami detecting. And type 3 detects sour. So now that we've gone over structure and function of both smell and taste, let's tie them together with some interesting facts. So, first off, olfaction is actually tied closely to emotion and memory. I discussed this a little bit earlier. Smells have a direct pathway to the limbic system, which is responsible for emotional processing and memory storage, which is why something like the smell of fresh baked cookies might transport you back to childhood, or why the smell of a perfume or cologne can remind you of the first date that you had with a guy in high school. Other senses don't have this strong direct connection, which is something that makes olfaction quite unique. Next up, no two people smell odors in the exact same way. So, genetic differences in olfactory reception means that the same odor molecule can actually be perceived completely differently by different people. So, for example, I think we all know this one. One person might think cilantro tastes fresh, but another person thinks it tastes like soap. I myself think it tastes like soap. And this difference comes down to variations in olfactory and taste receptors. So, kind of like a fingerprint in that way, no two people smell exactly the same. Some argue that there may be more than five sensations of taste. So, beyond sweet, sour, salty, bitter, and umami, research also suggests that we might detect fat as a distinct taste in the way that it coats the mouth, like ice cream or something like that. Other candidates being studied in Asian countries include the sense of calcium taste and metallic taste, although evidence is still coming out about that. They're not really sure on that yet. The exact mechanism for salt detection is still being studied. I mentioned this a little bit earlier as well. While humans seem to have a single main pathway for detecting salt, mice appear to have two, which might highlight how different species may adapt their sensory systems for survival. The sensation of flavor is actually a combination of taste and smell. So when we chew food, volatile molecules travel up through the nasal cavity, and we just learned about the process of olfaction. Well, this would be called retronasal olfaction. So this means that much of what we consider taste is actually smell working side by side with our taste buds, which is why eating with a plugged nose, if you have a cold, or during COVID when people lost their sense of smell, it can make food seem boring or even flavorless. With all these connections in mind, let's summarize by discussing the big picture. So, in total, smell starts with olfactory epithelium, travels through cranial nerve 1, the olfactory bulb, tract, and cortex, while taste begins with dissolved substances activating taste receptor cells inside of taste buds. Together, these two senses not only help us enjoy food, but also link directly to memory, emotion, and survival. Without them, eating and experiencing the world around us would be much less satisfying and much less fun. Finishing off with just a little works cited here. Thanks for listening. I hope you learned something.